Hi everyone, this is the last part of the digestive system lecture. So let's take a look. We're continuing with our little tour that we're taking here. So let's take a look at the small intestine. And we look at the gross anatomy of the small intestine. We find that it is the major organ of digestion and especially of absorption. So it's somewhere between 7 and 13 feet long from the pyloric sphincter to the ileocecal valve, which is where it joins the large intestine. It's relatively small in diameter, so it's an inch to an inch and a half in diameter. So it's like one of my pens that I use up on the board, one of those Expo pens. It has three subdivisions: the duodenum, which is mostly retroperitoneal. It's about 10 inches long. This is that first little piece off of the stomach. It curves around the head of the pancreas, and it has most features that we talk about. The jejunum, which is another 8 feet long, which attaches posteriorly by the mesentery, and then finally the ileum, which is the final 12 feet, which attaches posteriorly by the mesentery, and it joins the large intestine at the ileocecal valve. Some textbooks have already started putting the jejunum and the ileum together as one structure. And the reason they do that is because from the outside, there's not any real clear way of telling where you are. Now, histologically, looking at the inside, you can see some differences. So you will sometimes in a book see what's called jujoileum, and that means that they've put those two together as one piece. But for our purposes, we're still leaving them separate. So there is the picture we've been looking at all along. So we're talking about all of the stuff that's coiled up inside of the large intestine. Taking a look at the relationship between the liver, the pancreas, and the duodenum. So you see that first little, and the gallbladder as well, and that first little piece right there is what we call the duodenum, that first little 10 inches. The blood supply to the small intestine is supplied by the superior mesenteric artery coming off of the abdominal aorta. The veins that are carrying the nutrient-rich blood from the portal system drain into the superior mesenteric vein and then into the hepatic portal vein and then finally into the liver, as we already saw in circulatory system. The nerve supply, the parasympathetic innervation via the vagus nerve, sympathetic innervation via the thoracic splanchnic nerve, just like we saw earlier in the digestive system. So the circular folds, these are permanent folds. I call them speed bumps. They force the chyme to slow down as it's going through the lumen, and it allows more time for nutrient absorption. You have more circular folds in the duodenum and fewer as we get into the jejunum, and then finally very few when we get into the ileum. I read somewhere once that you have approximately 1,000 of those. The villi are the finger-like projections of the mucosa. So the mucosa is convoluted. There's a core in the very middle of that that has a capillary bed and a lymphatic capillary called a lacteal that's absorbing the fat. So remember, fat doesn't go through the hepatic portal system. Fat goes directly into the blood supply. And then there's the microvilli. So the villi actually have their own villi, which are extensions of the mucosal cells that give it a fuzzy appearance, and we call it the brush border. It's in that brush border that we have lots of membrane-bound enzymes that are used for the digestion of carbohydrates and proteins. So there's taking a look at that small intestine. You can see those ridges, those speed bumps. Those are the circular folds, and those are permanent. So we've seen ridges and expansion pleats like rugae already, but these are permanent. These don't stretch out and flatten out or anything like that. These are permanent. And then you see the villi, and then you see that the villi have their own villi, or the microvilli. There's a close-up of a villus. And so you see in the middle there is that green structure, that's the lacteal, and the lacteal is absorbing the fat off of the digestive tract after it's been emulsified by the bile. Taking a look at that villus when we are looking at it from a microscope, and then from an electron microscope. So they really are convoluted like that. So let's talk about the histology of the small intestine wall. There are modifications of the mucosa and the submucosal layers of the small intestine, and that is a direct reflection of the function in digestion or breaking things down. You have intestinal crypts. These are tubular glands that are scattered between the villi. There's 5 types of cells that you find in the villi and in the crypts. So you have the enterocytes. These make up most of the epithelial tissue that lines the inside of the tube. So it's simple columnar absorptive cells that are bound by tight junctions and contain a lot of microvilli. The function of the villi is to absorb nutrients and electrolytes. And again, the crypts, which are kind of like the indentions in between, this is what provides or produces the intestinal juice, which is a mixture of mucus and acts as a carrier fluid for the chyme. You will also find goblet cells. These are cells that are secreting mucus. They're found in the epithelial uh, tissue of the villi and the crypts. You'll find enteroendocrine cells. I don't know why all the numbering is all funny on this, but nonetheless it is. You'll find enteroendocrine cells. These are the source of enterogastrins, like hormones like CCK and secretin. They're found scattered in the villi, but there are some in the crypts. You're going to find Paneth cells. These are found deep in the crypts, and these are specialized secretory cells that are used for the immune system. And they're secreting an antimicrobial agent called defensin or lysozyme that helps to destroy bacteria. Again, your food is not sterile, so we don't want any bacteria that's in your food, if it's managed to make it through the stomach and the harsh environment in the stomach, we don't want that bacteria to pass over the digestive lining. So we've got these Paneth cells that take care of that. And then there are stem cells that are continuously dividing to produce all of the other cell types. The villi renew themselves about every 2 to 4 days. The mucosal associated lymphoid tissue, we talked about that back in the lymph chapter. Remember, that's called MALT. So it included some of the digestive system, and that's helping to protect against microbes. Also, you have individual lymphoid follicles that are in there. You have Peyer's patches. We talked about Peyer's patches in the past. These are similar to tonsils, if you want to think about them that way. They're aggregates of lymphoid nodule tissue. They're located in the lamina propria, so they're not on the surface. They're embedded in the actual wall, and their job is to form like this gauntlet of lymphoid tissue or immune tissue that the material that's passing over to the blood has to pass through, or over to the lymph, has to pass through so that it can be checked for any microbes. Again, we don't want to get microbes off our digestive tract into our blood supply or our lymph supply. The place where you're going to find the most Peyer's patches is at the distal end of the small intestine. The bacterial numbers have increased greatly at that point. Why? Well, there's been time has gone by, and bacteria multiply every 30 minutes or so, and so that food has been in your digestive tract for a while now, so the bacteria has had an opportunity to multiply. It's in the perfect environment. It's like it's in an incubator, so it's in the perfect environment to multiply. The lamina propria also has a lot of plasma cells that are secreting IgA, one of your immunoglobulins. The submucosa is made of areolar tissue, and in there you'll find duodenal glands. These secrete an alkaline mucus that helps to neutralize the chyme as well, because remember that chyme is coming out of the stomach with a pH of about 1, so it's going to take a lot of adjustment to make sure we get that back up to a neutral pH. Chemotherapy. We've talked about chemotherapy before. We know that it targets rapidly dividing cells, and hopefully that's going to get the cancerous cells, because that's one of the hallmarks of a cancerous cell is that they are rapidly proliferating cells. But unfortunately, one of the collateral damage events that happens is that we end up killing some of the rapidly dividing cells that do that normally, like the cells that are in the GI tract. So a lot of patients, when they're undergoing chemotherapy, have nausea and vomiting and diarrhea, and they don't feel good. And part of that is, unfortunately, the outcome of the chemo. What about the intestinal juice? So 1 to 2 liters. So I've lost count now about how many different liters we've put in, but we're way up in the 5 or 6 range of liters of fluid that we have secreted into the tube so far. So another liter or 2 every single day in response to distension or irritation of the mucosa. The major stimulus for production is hypertonic or acidic chyme. Intestinal juice is slightly alkaline. It's isotonic with your blood plasma. It's mostly water, but it does have some mucus in it, and the mucus is secreted by those duodenal glands as well as the goblet cells. So the chyme from the stomach has got partially digested carbohydrate, because remember that started up in your mouth, and it's got proteins that we started breaking down in the stomach, and we have undigested fat. So nothing's really happened to the fats so far. So just as a review, carbohydrate digestion starts in the mouth, it stops in the stomach, and it starts again in the small intestine. Protein digestion begins in the stomach and finishes in the small intestine. Fat digestion doesn't begin until we get to the small intestine. So kind of make a little note of that idea of where those different macromolecules are digested. The nucleic acids are also digested at the small intestine. We're just not getting any caloric value off of those. So it takes 3 to 6 hours in the small intestine to absorb all of the nutrients. So it took about 4 hours to empty your stomach. Then it takes another, let's just say, an average of 4 hours to get it through the small intestine. So we're now at 8 hours since you ate that meal. The sources of the enzyme for digestion in the small intestine are the bile, the bicarbonate, the digestive enzymes, but not those brush border enzymes, the ones that are found on the microvilli or in the microvilli of the villi of the small intestine. As these are, those are going to be imported from the liver. Not the brush border, but everything else is going to be imported from the liver and the pancreas. The brush border enzymes are bound to plasma membrane, to the plasma membrane of the microvilli, and they perform the final digestion of the chyme. So we regulate the chyme entry. We talked about this already. The chyme is being put in a very small amounts, a tablespoon or so at a time is entering the duodenum. It's usually hypertonic. The chyme delivery has to be slow. We want to stop any type of or prevent any type of osmotic water loss from the blood. The pH of the chyme is very low. It has to be adjusted so that it goes up to a neutral amount. It needs to be mixed with the bile and the pancreatic juices to continue the breakdown or the digestion of those macromolecules. The enterogastric reflex and the enterogastrones are controlling the movement into the duodenum to make sure that it isn't overfilled. After a meal, segmentation is the most common motion of the small intestine. So we had peristalsis that was moving stuff all the way through the stomach. But when we get to the small intestine, now we're talking about the process of segmentation. So this is initiated by intrinsic pacemaker cells. We talked about that already. It mixes and moves the contents towards the end of the small intestine, which is where we'll find the ileocecal valve. The intensity is altered by long and short reflexes and by hormones. The parasympathetic side of your nervous system increases the motility, and the sympathetic decreases it. Again, back to what we've been saying all along. Typically, the sympathetic side of your nervous system speeds systems up. However, the digestive, the urinary, and the reproductive systems are actually slowed down in the sympathetic system. Parasympathetic usually slows all your systems down, and those same 3 systems that we talked about just now, those are actually sped up by the parasympathetic. So think about the logic of that. If you are in parasympathetic mode, in other words, you're relaxed, you're not in any type of state of alarm or anything, this is a great time to spend energy to digest your food. If you're fleeing for your life, this is a terrible time to spend energy to digest your food. Between meals, peristalsis increases. It rises. It's initiated by the rise in the hormone motilin in the late intestinal phase every 90 to 120 minutes. Each wave starts distal to the previous wave, and it's referred to as migrating motor complex. The meal remnants, the bacteria, the debris, anything that we can't absorb, basically is going to move towards the large intestine. So the complete trip from the duodenum to the ileum takes approximately 2 hours. So we've seen this slide before. This is peristalsis, where we're just moving the bolus along, and then segmentation, where we're churning that bolus in place so that we can expose it to all of the different digestive enzymes to help break down all of it. So we're down to the very end of the small intestine. So we're at the ileocecal valve, and so they refer to this as a valve or a sphincter. It relaxes and starts to admit what's left of the chyme into the large intestine. The gastroileal reflex is what enhances the force of segmentation in the ileum, and gastrin helps to increase the motility of the ileum. The valve flaps closed when the chyme is has passed through, and it exerts a backward pressure. All sphincters, all valves, regardless of where they are in your body, their main function is to prevent regurgitation or backflow. So once we have sent what's left of our food into the large intestine, we do not want that backing up and coming in back into the small intestine. So just showing you the control of the small intestine motility there. And this is an overview of the function of the gastrointestinal organs that we've looked at so far. So you can look at the major function of each of those organs and then some comments and additional functions over on the side, kind of as a review to help summarize this. So there's your small intestine. We haven't talked about the large intestine yet, but there's the small intestine. You can see that its major functions are the digestion and absorption of the nutrients. So that lets us get to the large intestine, the end of the road here. So the gross anatomy of this structure, it has three unique features that we didn't see anywhere else. It has these things called tenini coli, which are bands of longitudinal smooth muscle that's found in the muscularis layer. It looks kind of like a ribbon coming through the middle of it. It has these pouch-like sacs that are called the haustra, and they are caused by the tone in that smooth muscle of the tenini coli. So it kind of like makes it bunch up a little bit. And then we have epiplotic appendages, which are fat-filled pouches of the visceral perineum that are kind of hanging off of the intestine. The intestine has several different subdivisions. The large intestine does. The first portion of it's called the cecum. Coming off the bottom of the cecum is the appendix, which is a mass of lymphoid tissue. It is part of the mucosal associated lymphoid tissue of the immune system. It is a bacterial storehouse that's capable of recognizing or recolonizing the bacteria in your gut if necessary, if you lose it all. So it holds on to some of the bacteria, and it has a twisted shape, and unfortunately that makes it very susceptible to blockages, and if that happens then the appendix enlarges and it can rupture. The colon, or the large intestine, has several regions, most of which are retroperitoneal. The exceptions to that are the transverse and the sigmoid regions. So when we look at the cecum, we see the cecum on the right-hand side, then the colon goes up, and that portion is called the ascending colon. So it's going to travel up to the level of your right kidney, and then it's going to hang a right-hand turn, and that's called the right colic flexure or the hepatic flexure. So if you say colic, you need to say right or left. If you say hepatic, it's assumed that your liver is on the right-hand side. Then it becomes the transverse colon, which travels across the abdominal cavity, and then it hangs another right-hand turn there. That one's called the left colic flexure or the splenic flexure. Same rule applies there. If you're going to refer to it as the colic flexure, you have to differentiate between right and left. But if you're just going to call it the splenic flexure, it's assumed that your spleen is in the right place on the left side, so there's no need to differentiate that. Then it heads down the left-hand side of your body, and that portion is called the descending colon. And then it makes an S shape at the end of it that travels down into the pelvis. That's called the sigmoid colon. And then the rectum are three rectal valves that stop any waste material from being passed with gas. The anal canal is the last segment of the large intestine, and the opening to the outside of the world is called the anus. There are two sphincters that are controlling anything from going out. The internal anal sphincter is smooth muscle. The external is skeletal. So it's the skeletal muscle that's voluntary, and so it's the external that we train little ones when we're potty training. So again, taking a look at the whole thing, they've removed the small intestine from the middle there. So you can kind of see that the large intestine sort of picture frames our small intestine, and you can see all those different parts that I was just talking about. You can see that tenia coli, which is the ribbon, the white ribbon, kind of grayish white ribbon that you see running through the middle of the whole thing. This is taking a look at the very end. So we're looking at the rectal valves and the external and internal anal sphincters. So what is the relationship to the large intestine to the peritoneum. So the cecum, the appendix, and the rectum are all retroperitoneal. The colon is also retroperitoneal except for the transverse and the sigmoid portions. The intraperitoneal regions are anchored to the posterior abdominal wall by mesentery sheets that are called the mesocolons. And so this is a great picture. We hadn't really seen a picture yet. Now, see that yellowy kind of looking thing that's laying on top of all of it? That's the greater omentum. I mentioned that when we were talking about this earlier in the lecture. That is the structure. It's like a sheet of fat, an apron of fat that's lying over top of all of your organs. And so this is just showing you the greater omentum, but it's also showing you the mesenteries. Better look at the mesenteries, the lesser omentum and the greater omentum. Now they've retracted that greater omentum, so you can see the transverse mesocolon, the sigmoid mesocolon, and each of those areas has a different name. We just collectively call it the mesentery. Looking at it from a side view to get the idea of what's peritoneal and what's not peritoneal or intraperitoneal, and then the location of those different organs and mesenteries. An appendicitis is an acute inflammation of your appendix. It usually results from blockage by fecal material that gets trapped inside of the appendix, and there's lots of bacteria in there. Venous drainage can be impaired, and that leads to tissue death because it doesn't get any blood supply, and a ruptured appendix can cause peritonitis that we talked about earlier. Symptoms of it is pain in the umbilical region, so usually right in the middle that moves to the lower right abdominal quadrant. You have a loss of appetite, nausea, vomiting sometimes, and then, of course, what they have to do is remove that. Typically, they can remove that. If it hasn't ruptured, they can remove that with a laparoscope, and it's a real quick and simple procedure. I think it takes all of about 5 minutes to do. The large intestine has a thicker mucosa when we look at it from the microscopic view. It's made of simple columnar epithelium, with the exception of the anal canal where it turns back into stratified squamous because of the abrasive nature of waste material. It doesn't contain any circular folds or villi or digestive secretions because we're all done with that by the time we get to the large intestine. It does contain lots of deep crypts that have a lot of mucus-producing goblet cells to help move things along. What about the bacteria? You have 1,000 different types of bacteria in the large intestine. They outnumber our own cells 10 to 1. So you have an enormous bacterial population that is living in the large intestine, and they are supposed to be there. They enter from the small intestine or from the outside world to colonize the colon. That often happens during a regular vaginal birth. And so one of the problems with a C-section is the process of colonizing the bacterial flora of the intestines is limited because the baby didn't pass through the birth canal and become exposed to the bacteria that are going to help it along the way. The metabolic functions of these bacteria are fermentation. So what do? They ferment the indigestible carbohydrates, and all of the mucin. And unfortunately, they also release irritating gases and acids when they do that. So how much gas do you release a day? In case you ever wanted to measure this and know this, it's approximately half a liter. So about half a liter. Don't know how they measured that. Not real sure I want to know how they measured that, but I'll leave that to your imagination. One of the other things your bacteria do for you is they produce vitamins. So they synthesize B complex and some of your vitamin K that's needed by your liver to make all your clotting factors. One of the other really important things that the bacteria that are supposed to be in your large intestine do is they keep the pathogenic, or the ones that can make you sick, they keep those pathogenic bacteria in check. So the beneficial bacteria outnumber the pathogenic and they suppress them. The immune system destroys any bacteria that try to come across the mucosal barrier, so you don't have to worry about the bacteria that's in there trying to sneak its way in. The immune system is very strong in that area. You have epithelial cells that recruit the dendritic cells to the mucosa to sample the microbial antigens and present those to the T cells in MALT and mucosal associated lymphoid tissue, which triggers the production of IgA and restricts those microbes from moving across the digestive lining into the blood supply. Gut bacteria and health, there's a lot of evidence out there that's starting to show that the kinds and proportions of your gut bacteria has a big influence on your body weight, on your susceptibility to various diseases, including diabetes, atherosclerosis, and fatty liver disease, and your mood, surprisingly enough. So when your bacteria are happy in your large intestine, you are happy. So maintaining your gut bacteria has become a pretty common routine health strategy for a lot of people. They make sure they eat yogurts and fermented things and kefir and all kinds of different products, sauerkraut, home brewed things like ginger ale and root beer. Surprisingly enough, all those kinds of things like that, not what you buy at the grocery store, but the stuff you actually ferment in your home, kimchi, all those kinds of things. They have lots and lots of bacteria in them, and they're actually healthy bacteria, and they apparently help you maintain a better lifestyle. Antibiotic-associated diarrhea accounts for approximately 14,000 deaths per year. Clostridium difficile, or C. diff, is an anaerobic bacterium that most of us carry in our intestine. It is the most common cause. When other bacteria are wiped out by antibiotics, the C. diff can flourish, and it causes pseudomembranous colitis, which is inflammation of the colon, and it can lead to bowel perforation and blood poisoning. So it's a really, really serious thing when someone has become infected with C. diff. So what's going on as far as digestion in the large intestine? So the product that is in the large intestine stays in there once it gets there for somewhere between 12 and 24 hours. There's not any more food broke down that's going to occur except what the enteric bacteria digest. But vitamins are being made by some of the bacteria in your large intestine. Mostly what's happening is water and electrolytes, especially your sodium and your chloride ions, are being reclaimed. So that's the main function, is to reclaim all that water. Remember all that water we were adding up? So we were at like 6 or 7 liters of water that we've added to the digestive tube. Well, if we don't reclaim all of that, then we got a problem, don't we? We can dehydrate out. That's what diarrhea is. Diarrhea is failure of your large intestine for some reason to reclaim all of that water. So another function is the propulsion of the waste material so that we can eliminate it. The large intestine is important for our comfort, but surprisingly not, it's not necessary for life. If your colon is removed, the terminal ileum is brought through the abdominal wall, and they make an ileostomy, a colostomy essentially. So what's moving things in the large intestine? You have haustral contractions, so those pouch-like structures. This is what's creating most of the contractions in the colon. They sequentially contract in response to distension or something being in there that's causing it to expand. These are slow segmenting movements, mostly in the ascending and the transverse colon. The gastrocolic reflex is initiated by the presence of food in the stomach. So you see this most apparently in babies and puppies. If you put something in one end, something comes out the other end almost immediately. If you've ever had a puppy, you feed that puppy, and as soon as that puppy's done eating, you have to take that puppy outside immediately. And the same thing's true with a baby. You feed a baby and you're changing a diaper right after that. Okay, that's called the gastrocolic reflex. It results in mass movements, which are big, slow peristaltic waves that activate 3 or 4 times a day and really push things through. So the large intestine has these mass movements. We didn't see mass movements in the small intestine. The descending colon and the sigmoid colon act as a storage reservoir for the waste material that we're about to eliminate. Low fiber diet can narrow the colon and cause strong contractions that increase the pressure on the walls, and that can result in herniations of the mucosa, what we call diverticuli, diverticula. So diverticulosis is the presence of these herniations. Common in the sigmoid colon. People over the age of 70, more than half of them have this particular condition of diverticulitis or diverticulosis. Diverticulitis is inflamed diverticula that rupture and leak into the peritoneal cavity and can actually be life-threatening. Another common condition you see is called irritable bowel syndrome. This is a functional GI disorder, and so it's kind of like it's like a, uh, it's like either you have diarrhea or you have constipation. So it's like these two things going competing with each other all of the time. So you have recurring abdominal pain, changes in the bowel movements, bloating, flatulence, which is gas, nausea. It can be depressing because, 1 minute you have diarrhea, the next minute you're constipated. It doesn't feel good at all. Stress is considered the common precipitating factor for this. So stress management is a really important treatment for irritable bowel. The process of defecation, mass movements are going to move the waste material towards the rectum. Distension is going to initiate a spinal defecation reflex. This is under parasympathetic control, so it stimulates contraction of the sigmoid colon and the rectum, and it relaxes the internal anal sphincter. Consciously, though, we have to relax the external anal sphincter. So again, that's what you're teaching little ones when you're potty. The muscles of the rectum contract to expel the feces. This is assisted by the Valsalva maneuver, which is the closing of the glottis, basically pushing down, contraction of the diaphragm and the abdominal wall muscles that cause an increase in the pressure inside the abdominal cavity and helps to push the bowel movement out. So showing you the defecation reflex. So this is controlled through the spinal cord. So you can imagine when somebody has an injury to this part of their spinal cord, they can't control this. Diarrhea is watery stools. It's when the large intestine didn't have enough time to absorb the water for whatever reason. It can include irritation of the colon by some bacteria. It can be from moving the digestive viscera, jostling around like in a marathon runner. Prolonged diarrhea can be a really serious condition. It can result in dehydration and electrolyte imbalance. So it causes you to go into a condition of acidosis and lose your potassium. So in the very young and the very old, diarrhea is a really big concern. Constipation is the other end of the problem, right? This is when the food stays in the colon for too long, and basically we pull too much water out of it, and so we can't get it to pass. So this can be from insufficient fiber or fluid in your diet, improper bowel habits, lack of exercise, or abusing laxatives. So people who abuse laxative continuously, their bowel becomes dependent on that, and they no longer creates the movements and the reflex that it needs to eliminate. All right, part 3 of this lecture is on the mechanisms of digestion and absorption. So digestion is the process that we use to break down food that we take in into those chemical building blocks. So just as a reminder, the chemical building blocks of carbohydrate are monosaccharides, and the chemical building blocks of proteins are amino acids, and the chemical building blocks of a fat or lipid is glycerol and fatty acid, and the chemical building blocks of a nucleic acid are the nucleotides. Those building blocks are the only things that are small enough to actually pass over the digestive lining and into the blood or the lymph. So digestion is a catabolic process that breaks macromolecules into the monomers, the individual subunits that are small enough for absorption. So there are both intrinsic and accessory gland enzymes that are involved in that. I've been showing you that all along the way. The enzymes carry out hydrolysis, meaning that water is added to break a chemical bond. Absorption is the process of taking those substances and moving them from the lumen of the tube into the body, either the blood or the lymph. So you have tight junctions that ensure that the molecules pass through the epithelial cells rather than between them. So the nutrients are actually traveling through the cells that are lining the tube. The materials enter the cell through the apical end or the lumen side, and they pass and exit out the basolateral side or the blood side. Lipid molecules can be absorbed passively through the membrane, but other polar molecules are using active transport to get from one side to the other. Most of the nutrients are absorbed before the chyme actually reaches the ileum. So let's talk some specifics here. The digestion of carbohydrates. Only monosaccharides can be absorbed, so we have to get down to the monosaccharide, the building block level. So starch and disaccharides are broken down to oligosaccharides and disaccharides. That process starts in your mouth with the salivary amylase that's in your saliva. The further breakdown into glucose, maltose, and sucrose has to happen, and then you have the final breakdown into monosaccharides, which are glucose, fructose, and galactose. So the point to this is it is a step-by-step process. It doesn't happen just in one event. It's breaking down into smaller and smaller and smaller pieces each time. So I like these charts that are in here because they help you understand how each of our macromolecules are different in this process. So this one is for carbohydrate digestion, or how do we break down carbohydrates and how does that process happen? The step of starch digestion in your digestion. Pancreatic amylase is going to break down starch or glycogen that has escaped the salivary amylase and is going to break those into the oligosaccharides or disaccharides. The brush border enzymes, which are lining the inside of the intestinal tube, those enzymes like dextrinase, lactase, glucoamylase, maltase, and sucrase further break them down into lactose, maltase, sucrose, and then they're broken into the monosaccharides, either glucose, fructose, or galactose. Monosaccharides are co-transported across the apical membrane of the absorptive epithelial cells, usually using secondary active transport along with sodium. So if you remember, secondary active transport is when you have a gradient that's generated and energy that's generated because of active transport, and then you have some things that move along with it, kind of like hitchhikers. So when sodium moves, the monosaccharides move along with them. Monosaccharides exit across the basolateral end or the bottom end by facilitated diffusion, meaning they need a helper to get out of the cell. So there's what they're showing you, the carbohydrate digestion absorption in the small intestine, how that whole process happens up at the top with pancreatic amylase, and then the brush border enzymes that are located in the microvilli and villi of the intestinal tube, and then how it's broken into monosaccharides, and then ultimately how those monosaccharides are leaving. People with lactose intolerance aren't able to consume anything with lactose in it, so they have deficient amounts of lactase. So any lactose that's eaten, any milk sugar that's eaten remains undigested, creates an osmotic gradient in the intestine, which keeps the water from being absorbed, and it results in diarrhea. So we aren't able to absorb the water back off of it. It can also pull water from the intestinal space and the intestinal lumen, so it creates a lot of extra water. Bacterial metabolism of undigested solutes that produce large amounts of gas result in bloating and flatulence and cramping pain. So the treatment for lactose intolerance is to add lactase enzyme drops to the milk or take a lactase tablet before consuming any dairy products. So what about proteins? How do we break those down and absorb those? So when we think about protein, our source is not just what we consume, but it's also the digestive enzymes and proteins from breakdown of the mucosal cells. So we're constantly breaking down the mucosal cells in the digestive system, and those are also a small source of protein. So polypeptides are first uncoiled, or proteins are first uncoiled, and then they're chopped into large polypeptides, and then they're chopped into smaller polypeptides, and then to small peptides, and finally they are reduced to individual amino acid monomers, and you have the occasional dipeptide or tripeptide, meaning 2 or 3 amino acids that are linked together. Digestion begins in the stomach when the pepsinogen is converted to pepsin because the pH is nice and low because of the hydrochloric acid. Once that chyme with protein enters into the duodenum, the pH level of the duodenum inactivates the pepsin. So here's the chart on this particular one for protein digestion. We go from a protein all the way down to the amino acid. We see that it starts in the stomach, it goes on in the small intestine, and continues in the small intestine. So the step of protein digestion in your intestine, the pancreatic proteases trypsin and chymotrypsin cleave the protein into smaller peptides, while carboxypeptidase takes off one amino acid at a time from the end. The brush border enzymes, aminopeptidases, carboxypeptidases, and dipeptidases, break the oligopeptides that were created in the dipeptides into their individual amino acids. So again, it's a stepwise process. The amino acids are co-transported across the apical membrane of the epithelial cell using secondary active transport carriers like sodium ion or hydrogen ion, and the amino acids exit from the basolateral side also using facilitated diffusion. So there is the whole picture of how protein digestion occurs and the breakdown of each of the individual steps. In rare cases, intact proteins are actually taken up by the intestinal epithelial cells by endocytosis and are released into the body. So typically that doesn't happen because we got to get it all the way down to the amino acid, but sometimes it doesn't get cleaved up. This is more common in newborn infants because of the immaturity of the lining of their intestine. So it may result in food allergies as the immune system sees that intact protein as antigenic or foreign and mounts an attack. Allergies usually disappear after the mucosa mature. What about lipids? So the steps in lipid digestion are a little bit different. We have to have emulsification, which is when we are going to mix the fat with some product, in this case the product is going to be bile, to allow the two to mix together. So the triglycerides and the breakdown products, of course, are insoluble in water. You know that oil and water don't mix, so they have to be pre-treated just like you pre-treat your laundry when you've got stains and oil and things on them. That's basically what bile is. Bile is just a pre-treated detergent that's going to break the large fat globules into a smaller one. Then digestion has to occur, and that's going to happen because of the pancreatic lipases. They're going to break down the fat into monoglyceride plus 2 free fatty acids, and it's going to produce, they're also going to produce micelles. Micelles are little teeny tiny beads of fat. So this is the product that we end up with when we break the fat all the way down. We coat those with bile salts and lecithin, and then the fusion process happens where the lipid products are going to leave the micelles and cross the epithelial membrane via diffusion. The chyromicron formation, the lipid products are converted back into triglycerides and packaged with lecithin and lipoproteins, and they form a chylomicron. And chylomicron transport occurs as they are exocytosed from the basolateral side, the base of the cell, and enter into the lymphatic lacteals, and they are eventually dumped into the venous blood of the thoracic duct. So the important thing to really get out of this is that fats are transported differently. Because fats are transported in the lymph and then dumped into the blood and don't run through the liver first like they do with carbohydrates and proteins, that's why we can have fatty buildup inside of our vessels, because if that went to the liver first, we would never have that problem. But because of the way that fat is absorbed and transported rather than using the hepatic portal system, there's that possibility. Once in the blood, the chyromicrons are broken into free fatty acids and glycerol by lipoprotein lipase, so they can be used by the cells. Short-chain fatty acids can diffuse directly across into the blood. All right, so fat digestion. So they show you how emulsified, in other words, it hadn't been mixed with the bile yet, triglycerides pass through the mouth, the stomach, small intestine. It's not until they're mixed with that bile in the duodenum of the small intestine, and then they meet up with the pancreatic lipases at the same place that they're broken down. Here's the picture showing you the emulsification, digestion, absorption of fat. So there's your giant fat globule up there at the top. The bile salts are going to break that apart, and they kind of make these little barriers around it. The fat droplets are coated, I guess is another way of saying it, with those bile salts. And then those are able to move across the digestive lining and directly into the lacteal. What about nucleic acids? So again, we don't get a caloric value off of nucleic acids, but we do take the DNA and the RNA from the cells that we consume. So as long as you're eating natural food, plants, animal material, it has DNA and RNA in the cells of that tissue that you're eating as well. Where you won't have any DNA and RNAs when you're eating frankenfoods, so Twinkies and all those chemical type foods that aren't actually real food, they don't have any DNA and RNA. So the pancreatic nucleases are going to hydrolyze the nucleic acid into the nucleotide monomers. The brush border enzymes, the nucleosidases and the phosphatases, are going to break the nucleotides down into free nitrogen bases. So that's the A, T, C's, and G's. Pentose sugar and phosphate ions. Those breakdown products are actively transported by special carriers in the epithelium of the villi. So here's how we digest our nucleic acids. Again, this happens because of enzymes coming from the pancreas and then the brush border. So what do we do with all those A, T, C's, and G's and all that stuff we break down? We use it to make new DNA. So it's, again, we don't get any calories off of it, but we do use it for building stuff in our body. What about the absorption of vitamins, electrolytes, and water? So vitamin absorption occurs in the small intestine. The fat-soluble vitamins are A, D, E, and K, and so ADEK is the term you use for that. And what it means, fat-soluble, means that they are carried in those micelles. They're carried by fat, and they diffuse into the absorptive cells. So they need fat to be able to be absorbed. Water-soluble vitamins are C and B, and they're absorbed by diffusion or by passive or active cotransporters. And so you don't need any fat for these. Fat-soluble vitamins we can store. Water-soluble vitamins we cannot. So if you take too many vitamins, the fat-soluble ones you can actually build up a toxicity to. The water-soluble C's and B's, you're just going to urinate those out. Vitamin B12, which is a large charged molecule, binds with the intrinsic factor from the stomach, if you remember that from a couple of lectures ago, and is absorbed by endocytosis. In your large intestine, vitamin K and B vitamins from bacterial metabolism are absorbed. How do we absorb our electrolytes? Most of your ions are transported actively along the length of the small intestine. So all along the small intestine, we're actively absorbing those electrolytes. Iron and calcium are absorbed specifically in the duodenum. Sodium ion absorption is coupled with active absorption of glucose and amino acid, meaning sodium uses glucose and amino acid. Those three kind of all work together. They move together. Chloride is transported actively. Potassium ion diffuses in response to osmotic gradients. It's lost if the water absorption is poor. And usually the amount that's in the intestine is the amount that's absorbed as far as potassium is concerned. Iron and calcium absorption is related to need. So the ionic iron is stored in the mucosal cells with ferritin. So iron is a heavy metal. It can't be free in the body, so it has to be stored with a helper, and then when it needs to be transferred, it's transferred with a transfer molecule called transferrin. So free iron is not allowed to happen in your body because it's a heavy metal. Calcium ion absorption is regulated by vitamin D and parathyroid hormone. So vitamin D, the process of making vitamin D starts at the skin with sunlight. And that works with parathyroid hormone. So if you've ever maybe looked at a jug of milk, you notice it says that it's got added vitamin D and calcium. So those two are usually together. What about the absorption of water? 9 liters of water. 9 liters of water, usually from the GI secretions that we've been talking about, are going to enter the small intestine. So 95% of that water is absorbed in the small intestine by osmosis. Most of the rest is absorbed in the large intestine. So we talked about that. The job of your large intestine is to reclaim any water that's left in the lumen of the digestive tube, or it's going to create diarrhea. Net osmosis occurs if the concentration gradient is established by active transport of solutes. So if solutes are moving and using energy, water will just tag along with it. Water uptake is often coupled with solute uptake, which is where we always say water follows salt. Water follows sugar. And so if the salt moves, the sodium moves, or the sugar, glucose moves, water typically goes with it. Malabsorption can be caused by anything that interferes with the delivery of bile so that you can't break down fats, or pancreatic juice, which means you can't break down any of your nutrients. So If you have damaged intestinal mucosa, like from a bacterial infection or from antibiotics, that can interfere with your ability to absorb your nutrients properly. Gluten-sensitive enteropathy or celiac disease is a common malabsorption disease. It's basically an immune reaction to gluten, which is found in wheat products, rye products, and oatmeal. And so the gluten causes immune cell damage to that brush border and to the intestinal villi, and the treatment for this is to take all of the gluten out of your diet. So really popular diet right now are these gluten-free diets. At the end of the day, what it really looks like, and the research is starting to demonstrate, is that we were never really meant to eat as much wheat as we do, and there is a lot of wheat in a lot of processed food. You may not even realize it has all kinds of names like modified something something or whatever, but it's got a lot of different alternative names, and humans weren't meant to eat that much wheat. We can eat some, but we weren't meant to eat that much. So there is a researcher at MUSC that postulates that most people have some level of gluten sensitivity, and simply taking some of the wheat out of their diet, or at least attempting to be cognizant of it, usually makes them feel better. So just something to think about if you're consuming a lot of bread products, a lot of rye, a lot of oatmeal. Cleft palate and cleft lip are the result of what happens when the palatine bone and the palatine process of the maxilla fail to fuse together. So those were separate bones during development, but they don't fuse together. So you have that split lip. This can be dangerous in the sense that it makes it so that the baby can't latch on. All right. A transesophageal fistule is an opening between the esophagus and the trachea, for obvious reasons. That's a huge problem. And cystic fibrosis is a genetic disease that produces this really thick paste that we think about typically with the lungs, but it also happens in the digestive system. So it can block the pancreatic duct and make it so that you can't deliver the enzymes you need to break down your nutrients. All right, that's it for this chapter. If you have any questions, be sure to let me know.
That is the end of the digestive chapter. So you are ready to study and take your quiz.